Tiwari. All right, so we're recording. Dr. Tiwari is an associate professor at the Amity University in Noida, India. She works in the Diatom Research Laboratory. And today she'll be talking to us about the role of diatom algae in carbon dioxide mitigation and generation of high value products. And she is one of the very few people that are working in this field. So let's enjoy and listen to Dr. Tiwari's talk today.
Thank you for such a nice introduction. I hope my slides are visible at your end.
Yes, we can see them.
So good morning, good afternoon, and good evening to people around the world. We are going to talk about the role of diatom algae in carbon dioxide mitigation and generation of high-value products. So first, I would like to introduce diatoms are one of the special classes of algae which are eukaryotic in nature, and we find them in almost every habitat. Diatoms are freshwater as well as marine, and they are photosynthetic in nature. They contribute towards 40% of global primary productivity, and they accumulate a lot of lipids in them, and we find their applications in diverse range of industries. So we can see here diatoms are of two types, centric and pinnate diatoms. So when we consider their unique morphology, the cell wall, that's an exception to other classes of algae. The cell wall is made up of silica. So the silica helps to absorb a lot of heavy metals, different types of contaminants. So silica, and also there's a very low energy requirement for silica buildup. So that's yet another conservation, natural conservation in part of diatoms. The vacuoles of diatoms are bigger compared to other algae, which are the site for storage of nutrients. And the major storage material is chrysolaminarin, which is a high value compound. Also, the lipid molecules or the lipid body is a response which they bear in the habitat which they live, and the lipid body, the number of lipid body, the orientation, the size changes according to the habitat wherein they live. The nucleus is very efficient, and also we find a combination of genes in diatoms, some of plant origin, some of animal origin, and some of bacterial origin. So that makes them metabolically very unique. The other cellular entities enhance them to have a higher photosynthetic efficiency, and also they have a complete citric acid cycle. So the role of diatoms in nature is one of the dominant forms wherein they sequester enormous amount of nutrients that includes nitrate and phosphate. So this nitrate and phosphate, which is generated due to a lot of anthropogenic interventions, different types of industries that is remediated efficiently by virtue of the different processes like biotransformation, bioabsorption, adsorption. So the diatoms in due course of time have devised different types of mechanism to combat the negative effect of anthropogenic pollutants. Till now, we have more than 2 lakh species of diatom reported from different types of habitat. And because of their metabolic efficiency, they can withstand a huge and a higher concentration of pollutants. Also, their morphology changes in response to the environment where they live. So they are excellent bioindicators. They are a reliable biomonitoring sources for water body. Also, they contribute towards the primary productivity, and they have a very fast growing rate. And also they have high sinking rate, which allows them to settle down at the bottom of the water body and make it nutrient rich. So there are various environmental applications of diatoms in nature. So this is the basic scheme of a diatom biorefinery, wherein we have generation of enormous amount of wealth from waste. So this waste includes different types of industry, agricultural waste, domestic waste, pharmaceutical waste, livestock, aquaculture waste. So different types of waste that gets into the water body. Since diatoms are the natural inhabitants of the water, they have this natural ability, inherent ability to quench a lot of pollutants by virtue of their metabolic potential. So diatom advantages in the refinery includes their amicability for mass cultivation, their ability to produce lots of lipids in response to the stress, and the biomass which is generated from this waste grown diatom. That biomass finds a lot of applications as biofuels, as nutraceuticals, the different pigments which have therapeutic value. Along with that, the biomass of the diatom also has biofertilizer and plant growth stimulating abilities. They can act as aqua feed for zooplanktons, ornamental fishes, and different classes of fishes. They are one of the most, digestible feed for fishes, even in the larval stage. When the stomach is not well developed, the diatom feed can be easily ingested and utilized by different fish larvae. So the methodology that we follow includes isolation of diatoms from different sources. That includes freshwater as well as marine. Also, we have the identification of diatoms on the basis of, initially we go ahead with their examination on light microscopy. Then further we go for scanning electron microscopy, and the validation is done through the molecular analysis of 18S RNA. And then once the pure culture is there, we tend to optimize the culture condition to enhance the biomass productivity. We administer the culture to different cultural conditions, which includes adaptive lab evolution, chemical engineering, and then screening the most promising high biomass producing diatoms. Further, the promising diatoms, they are harvested utilizing different techniques, and the lyophilized biomass is further utilized for their different applications. So isolation of diatoms from my country, we have isolated diatoms from diverse freshwater and marine habitats. And you can see here some of the valuable culture, which includes Melocera, Gomphoneema, Navicula, Nischia. So the strategy that we use for their isolation is, initially we fortify them with diverse concentrations of silica, and then regularly check their growth. Once the culture has initiated growing, then we tend to grow it at different concentration of nutrients and validate their media composition. So this is the identification of few native diatom isolates and their SEM images. Chitoceros gracilis, which is one of the very important strains for aquaculture application. We have isolated it from the southern coast of India, and its molecular identification was done on the basis of 18S RNA. These are few of the diatom species that we have isolated in our laboratory. So it's an everyday task to bring water samples and examine them, and then go ahead with their isolation. So I would also like to add here that one of the most tedious tasks associated with diatom research is pure culture isolation. Getting the diatom pure culture from the microbial population, from the green algae population, is perhaps one of the biggest research gaps that needs to be addressed. So when we grow the diatoms on wastewater, what they do is they utilize the CO2 that is present in the environment and the different types of waste like municipal wastewater, dairy wastewater, brewery, aquaculture wastewater, pharmaceutical wastewater, the nitrogen and the phosphorus ratio, which ranges from 10 to 17%, all that waste is utilized as nutrients for the growth of diatom algae. So what they are doing initially, they are utilizing the nutrients or along with the pollutants which is present in the water body and also the atmospheric CO2, mitigating it to oxygen and many valuable compounds which find their applications in different industries. So after the treatment or after the growth of diatom in that wastewater, theoretically we get a biomass which ranges from 0.3 to 41 grams. So that is the biomass conversion and the CO2 mitigation, which is leading to a high value compound. So the CNP ratio is also reduced. The water body becomes fit for the different aquatic organisms, both flora and fauna. And we have the dietary biomass generated from the waste, which is rich in carbohydrates, proteins, lipids, and nucleotides, along with the pigments. So it's a complete biorefinery sustainable system wherein we have valuable compounds generating from waste. So this is one of the experiments wherein we have utilized aquaculture water, because the aquaculture water is rich in different types of nutrient, phosphorus, ammonia. So this acts as a natural nutrient source for growth of diatoms. So we cultivated diatoms in different concentration of aquaculture water, and we could see that there was enhancement in the biomass production. Also, there was a drastic reduction in the nitrate and phosphate. And this aquaculture water, since it was fortified with nutrients, it triggered the metabolite production, which could be valuable as a feed and also as plant growth stimulators. So we can see the growth analysis of diatom in aquaculture water and the biomass productivity, which enhances due to the nutrients present in the aquaculture. So the percentage of nitrate and phosphate remediation, which occurred by virtue of diatoms, ranged from 60 to 90%, and there was also good amount of carotenoid production. So these carotenoids are high value compounds which have huge therapeutic value. Also, the lipid productivity was good. So this lipid could be utilized as a drug molecule or as a biofuel. Also, we have tried to remediate aquaculture wastewater using immobilized diatoms. So Nischia is one of the pollution tolerant species. So we have immobilized Nischia in calcium alginate and validated how efficient they are for wastewater remediation. And we could see that the remediation of nitrate, phosphate, and ammonia ranged from 60 to 90%. By virtue of the immobilization. So immobilization has its own advantages, wherein this compartmentalization helps the microbe to get a better habitat, to get a compartment wherein it can grow faster. So this development of alginate bead, the beads were stable, we have reported till 9th week, but the beads were stable even till 6 months. So we filed one patent related to the application of immobilized bead as a live feed. In addition to the aquaculture water, we have also investigated whether municipal wastewater can foster diatom growth, and we compared it with the ISO crisis. So we could see that diatoms also have a high growth rate in municipal wastewater, and the lipid composition was perfect for biodiesel production. So then we analyzed the lipid bodies grown on wastewater, and we could see that the lipid bodies, with the help of Nile red staining, we could see that the lipid body diameter and the volume can be calculated. And we can see here the conspicuous lipid bodies using Nile red staining. So the lipid body characteristics, the initial size and the diameter was determined. So this clearly validates that wastewater can be a very good nutrient source for growth of diatoms, and the lipids can be utilized for different applications. Also, the lipids can be used as biofuel, and the suitability of diatom-based biofuel includes the feasibility to cultivate them, and the percentage of lipid compared to the other algae is relatively higher. Also, this oil is capable of conversion directly. It can be processed to biodiesel. So it can be a good solution to reduce our dependence on fossil fuels. Also, it is eco-friendly and biodegradable. It's non-toxic, and the sulfur content is also not there. So this is the FAME profile for three diatoms, Chitoceros, Skeletonema, and Theocerocera, which shows that they have good concentration of saturated fatty acid and monounsaturated fatty acid, which makes them ideal candidate for biodiesel production. So the biodiesel quality that depends upon the iodine number and the cetane number, and the cetane number for the diatoms that we have investigated, it varies from 40 to 50%, which is quite promising. Skeletonema is a strain which has a very low iodine value, so that also is a positive factor which can be utilized further for biodiesel production on a larger scale. These are few of the publications that we have for biodiesel production. Another aspect is what are the different value-added products from diatoms. So they are a huge treasure house for different metabolites. So this metabolite includes carbohydrate like chrysolaminarin, carotenoids like fucoxanthin, omega-3 fatty acid, which have huge antimicrobial, anti-inflammatory, anti-cancerous effect, and also they have a range of sterols which can act as precursors of drug molecules. So diatoms, they are rich in EPA, DHA, different pigments like fucoxanthin, oxalipins, sterols, cellophans, polysaccharides, and diverse carotenoids, which have huge therapeutic value. Few bioactive compounds from diatoms include polyunsaturated fatty acids, carotenoids, EPA, DHA, also diatomaceous earth, which is a natural biocontrol agent, is also produced from diatoms. So the next perspective for the study is valorization of diatom biomass that includes the biochemical screening. So once we have the pure strains, then we go ahead with estimating their biomolecule efficiency. So we have estimated the fucoxanthin yield. So in Chitoceros, it ranged from 0.217 gram per liter, so which was quite promising. And the secondary metabolites, in case of a strain called Skeletonema, we could find that the highest phenol content, 86 grams per liter per dry cell weight, and different types of flavonoids like tocopherol were also high in case of skeletonema. The omega-3 fatty acid like EPA and DHA, when it was validated, Thiocera could, under natural conditions, the concentration varies from 25 milligrams per dry cell weight, which is quite promising. So in addition to it, we also did the metabolite profiling to see what are the different molecules and what can be the key target proteins. So the active molecules which we could identify using GCMS, that included momoinositol, phytol, and brassicastérol in different high-value compounds, some of which have not been reported in other classes of diatoms. So these molecules were identified with the help of GC in different types of diatoms. So Cylindrotheca amorpha, Thiocera, it could actually exhibit a diverse range of bioactive compounds which are yet to be investigated for their medicinal applications. So in plants, these compounds like phytols and momoinositol, phytol has huge antimicrobial, anti-cancerous, anti-inflammatory, anti-diabetic efficiency, while we find momoinositol is valuable in treatment of neurodegenerative diseases like Parkinson's, Alzheimer's. Also, these biomolecules, they play an important role as inhibitors, receptor molecules, and also as anti-inflammatory in nature. They are also reported to help in treatment of diabetes, lowering down the blood pressure. So that this fact clearly validates the huge research gap that exists and the potential which the diatom possess. So we screened few native diatoms for their EP and fucoxanthin production, and we could see a diverse range of production of EP and DHA from different types of diatoms. In addition to it, we also did few studies related to examining what stage is better for extracting which type of compounds. So we could see that in the stationary phase, the lipid productivity is the highest, while in case of pigments in the exponential phase, we could get highest amounts of pigments. Also, the different stages affect the productivity, the primary as well as the secondary metabolites which are present in diatoms. So optimizing the different stages wherein we get maximum productivity of a metabolite can also be a good tool to enhance their commercial application. Also, there are seasonal changes in the productivity, and summer months are good for metabolite production, while winters are excellent for lipid production. That might be that reduction or in temperature leads to a stress condition, which allows the diatom to produce copious amounts of lipids. So we could see the EPA production. EPA is a high value compound, and the vegan diet, it lacks EPA. So this way, investigating diatoms as potential source of omega-3 fatty acid can also be a step forward in developing a vegan-based omega-3 source. So the pigment profiling, when we did through HPLC, we could find that the chlorophyll concentration, it ranges from 4 to 5 milligrams per liter, while fucoxanthin was 18 milligram per liter, chlorophyll A 10.26 grams per liter, while beta carotene was 2.61. I would also like to add here that fucoxanthin has excellent anti-cancer properties, and till now, it has been reported in treatment of breast cancer, in leukemia, in lymphoma, in the treatment of cervical cancer, skin cancer, prostate cancer, bladder cancer, and different types of liver cancer. So investigating with the strains which have high concentration of fucoxanthin, as well as enhancing the fucoxanthin production through genetic engineering or metabolic engineering, can also be a step towards developing compounds or developing drugs for treatment of different types of cancer. So cultivating different diatom strains, we also compared the efficiency of diatom carotenoids compared to the blue-green algae, and we could see that the pigments which are present in the diatoms, these pigments, they are capable of inhibiting the growth of pathogenic microbes like Staphylococcus aureus, Klebsiella, and a range of fungi. Different highly pathogenic Candida species could be inhibited by the different carotenoid pigments which are present in diatoms, and also we could validate that these pigments can be utilized as antimicrobial agents. So screening for the bioactive potential in diatoms included examining their activities like lipids, the phenolic content. Phenolic content imparts the antimicrobial potential to different types of organism. So we could estimate that the highest phenol content was observed in Skeletonema, and also these strategies can be utilized for enhancing the productivity in diatoms. The flavonoid contents like tocopherol, the tocopherol content in case of Skeletonema, it ranged from 30 milligrams per dry cell weight of the species. So the tocopherol has a lot of medicinal value. So network analysis we did for few of the compounds, and this network analysis revealed that oleic acid and linerolic acid are the major ingredients, and they play a vital role in treatment of heart diseases, in lowering down the cholesterol, and in preventing different types of cancers. So the carotenoids, the mechanism that they follow is they reduce that helps in reducing the swelling and also in treatment of different types of tumors. The sterols, they have been reported in the treatment of breast cancer. The fatty acid, it helps to convert different types of enzymes called cyclooxygenases. So that helps in treatment. Also, the polysaccharide, the bound and the unbound polysaccharides, they have been reported in the immunomodulatory system. The different peptides from Nischia, Navicula, Pheodactylum, they have been studied as antioxidative molecules, and we have also estimated the antioxidative potential of diatoms. And we have seen a huge amount of enzymes which are produced in diatoms, which can be utilized on commercial platform. So utilizing the diatoms as aquafeed is yet another perspective. So to validate how suitable a particular diatom is as a feed, initially we isolate the microorganism, and then we see what are the culture conditions. Initially, we try to mimic the natural habitat of the particular diatom, and then what are the best conditions to optimize the metabolite production of the diatom, particularly the high carbohydrate content and high protein content is one of the desirable characters for as an aquafeed. So once we have done the mass cultivation for the diatoms, then what we do is we go ahead with the metabolite analysis. And then what we do is we go ahead with the phenol, the secondary metabolite estimation, then the lipid estimation is done, because high omega-3 fatty acid is something suitable for the fish consumption as well. So the antioxidative activity, because antioxidative activity, if the feed is rich in antioxidants, antimicrobial efficacy, that is good for the aquatic system as well. So high catalase activity suggests that diatoms can be a potential antioxidative source. Also, we have estimated enzymes like superoxide dismutase, peroxidases, and different antioxidative compounds were estimated. And we could also see that the antibacterial activity of diatoms could inhibit the pathogenic strains like Aeromonas, which is a common fish pathogen. So in the water body, if diatoms are there, they not only act as a good feed, remediate the water body, they also helps as a natural biocontrol agents in inhibiting the activity of different pathogenic bacteria and fungi. So it's a complete holistic system wherein we are generating wealth from waste. So the slide shows the lipid profile of few of the diatoms which we have validated as sustainable feed. So high content of EPA and DHA along with the carotenoids can validate or can vouch for diatoms and other strains like Isophysis as aqua feed. So the next stage is optimizing the mass cultivation. So for mass cultivation, we have tried with batch cultivation, continuous system, and fed batch system to optimize the biomass yield and then estimate the lipid productivity. So we could see that when nanosilica is utilized for the growth of diatom, that triggers the growth of diatom and also enhances the lipid productivity. So in conclusion, I would like to mention that they are unique organisms to be worked, and we can address the different challenges related to diatom research by isolating more species and then investigating their metabolic potential. Also, silica in the nano formulation can be a good strategy to foster diatom growth. And diatoms, they do have a good amount of sterol concentration, which needs to be explored. And this is one class of algae which needs to be investigated more and more so that we come up with their application, and we can also propose to have novel therapeutic compounds in the treatment of different incurable diseases, and also it can be a great milestone in treatment of cancer. So these you can visit our lab page for the different publications that we have. So in nutshell, what we are doing is try to develop a circular bioeconomy wherein generating all the valuable products from diatoms and investigating their applications as therapeutic molecules, as aquafeed, as plant growth stimulators. So that's my research team at Diatom Research Laboratory. Thank you so much.
Thank you very much, Dr. Tiwari, for this fantastic talk and opening our eyes on the hidden potential and treasures of diatoms. And so we have time for one very, very quick question. And if not, we can save questions towards the end, where we'll have ample time to ask more questions to both of our speakers. So any quick questions for Dr. Tiwari? Okay. But still save your questions because we'll have time at the end to address both speakers. And so thank you, Tiwari, for the talk.
Thank you.
So next we'll have Richard Mugani. He is a PhD student at the Cadi Ayyad University in Marrakech, Morocco, and he works in the Water Biodiversity and Climate Change Laboratory. And he's also a visiting researcher at the Leibniz Institute for Freshwater Ecology and Inland Fisheries in the Department of Plankton and Microbial Ecology in Germany. And today he'll be talking to us about the impact of microcystins on the dynamics of rhizosphere microbiota, a focus on extra and intracellular microbial enzymatic activities. So the floor is yours, Richard. Richard, you're still muted.
Thank you very much for your nice introduction. And now I'm going to share my screen. I hope you can see my presentation. So today I'm pleased to be here to talk about the impact of microcystins on aquatic and soil microbiota dynamics with a focus on extra and intracellular enzymatic activities. As you said, I am a PhD student in Morocco, Cadi University, and there I am supervised by Professor Brahim Udra and Professor El Khalofi Fatima, and here as a visiting researcher at Leibniz Institute, IGB, I am supervised by Professor Hans-Peter Grossart. My talk today, I will be outlined as follows. First, I'm going to introduce the impact of microcystins in water and also on cultivated plants. And then I'm going to shortly introduce the microbial enzymes of biochemical cycles. I'm going to stress the reason why there is this focus on these kind of enzymes. And then will come the testing process, the experiments we conducted to respond to the questions we posed. And after that, we will conclude. Yes, microcystins, there are some hypertoxic cyanotoxins that are produced by certain strains of cyanobacteria. And within we can find Microcystis aeruginosa, Planktotrix, Lyngbia, and most importantly, the species Microcystis aeruginosa is the one that is very well studied because of the deleterious toxins it secretes, which are microcystins, and also the impact on different organisms, either in water and also on human. So in Morocco, we have the Lake Takelkust, which is a dam that was created to produce water that is used for irrigation, and also the water that can be used for drinking purposes. Here, a focus or some stress is that microcystins that have been quantified in the lake goes up to a concentration of 95%, 95 micrograms per liter. But as you can see, the World Health Organization has fixed a guideline that drinking water for health issues cannot contain more than 1 microgram per liter. And the microcystins, why is it a focus on these cyanotoxins? We focus on that because it has deleterious effects on human body. Yes, of course, to date, there is no fatalities that has been recorded for people to have consumed microcystins. However, there is this study in Brazil that showed that 76 patients died in a hospital in 1966, when these people received dialysis with water that was contaminated with microcystins, with a concentration going up to 90.5 microgram per liter. And seeing this, you can somehow see that or make a link that the concentration of microcystins that can be found in Lake Takelkust, a focus of my PhD, is well beyond than this concentration of microcystins that caused the fatalities. And the organs that can be affected by the toxins can include lungs, liver mostly, and immune system, and also stomach, small intestine, and the colons. There are some other studies that show that microcystins can also somehow initiate or cause cancer to develop. So in agriculture, there is lots of studies that have shown the impact of microcystins on different aspects and on different organs of the plant, beginning by the aerial parts like the leaves or fruits, and also some parts under the ground like roots. And we see through different publications and literature that microcystins exactly cause problems on plants, and also the specificity of this toxin is that it can accumulate in edible parts of the plant and then make a problem to people who are going to consume it. However, even if these amounts of studies have shown the impact and the danger that microcystins can cause on plants, we still cannot say exactly what are the processes that can happen in this hampering method. Somehow, when these studies try to link the plants that have been grown in soil that are not sterile, and also soil that is sterile, we have noticed that plants that are grown in soil with microbiota, they are a bit protected to the effects of these microcystins. But the plants that have been grown in soilless media or in aquaponics, we know that microcystins are going to affect them greatly. So here, the focus of this talk today will be about the microbial enzymes that intervene specifically in biogeochemical cycles. And by that, we are talking about some enzymes like beta-glucosidase, which is an enzyme that somehow intervene in the making accessible some carbon in soil, which somehow is used by other organisms like microorganisms to maintain some other interactions with plants, and therefore the structure of the soil. There is also this N-acetylglucosaminidase, which is an enzyme that participates in mineralization of carbon, also nitrogen. Both are the elements that are very important to plants and also to microorganisms. And also comes the protease, which is also an enzyme that participates in the degradation of proteins, also making available nutrients to plants and also to microorganisms. There are phosphatase intervening in mineralization of phosphorus, and also another key point element that is very important in plants growth, and the aryl sulfatase, also another element that can intervene or that plants need. Urease, that is an enzyme that participates in a degradation of urea, which is the base of more chemical fertilizers, and also dehydrogenase, which is an oxidoreductase enzyme that that make available the energy in soil. So even if there is no understanding of what are the specific mechanisms that intervene for microcystins to cause the retardation of the growth of plants, these key elements, we do not know how much they are affected in the soil by microcystins, and if somehow microcystins can block or can prevent this enzyme to function properly, we can understand that the outcome on plants is going to be very serious. So for this, we isolated from, as you can see in picture here, let me take the pointer. So as you can see here in right, so the water of Latecoustis within blooms and Microcystis aeruginosa scum, we isolated the bacteria that can resist or tolerate a lot of concentrations of microcystins, and these bacteria, we test them for their capabilities to promote growth, like the production of phosphorus, the solubilization of potassium, and also the production of some phytohormone enzymes like auxin, and also the production of exopolysaccharides, which are the elements that can make a real difference in plant growth. And we inoculated this bacteria to pots in which we have grown pea, and we let this experiment running for 3 months until we had fruits. And after that, of course, the plants were irrigated by increasing concentrations of microcystins, beginning by 0 as a control, 50 micrograms per liter, and 100 micrograms per liter. And the experiment was run for 12 weeks, and we irrigated the plants 3 times with these concentrations, with these increasing concentrations. And after the maturation, we collected the soils, of course, within these treatments, and we analyzed the enzymes, the enzyme that we just mentioned, either by fluorometry or colorimetry. The results are very interesting, as we can notice that something I should mention is that we separated the experiments in 2 sets. The first, as you can see here, Ni is a sterile control, and we took 3 bacteria that were very tolerant to the concentrations of microcystins, and then we did a consortia to see exactly how these bacteria can interact with increasing concentration of microcystins, and then help the plant to sustain the concentration. And also here we see from the soil what really happened. And the second set, as you can see here, is also the control. that was not added by bacteria, but using a soil that is not sterile. And after, we also added the isolated bacteria in the same soil that is not sterile. And as you can see, there is a sharp decrease when we consider the control with 0 microcystins. You can see here in blue, and the 100 micrograms per liter. So we can see that there is a decrease in beta glucosidase, but we do not see this significant effect when we consider the control and 50 micrograms. This somehow tells that the microcystins has a great impact on the beta glucosidase enzyme in the soil. We can observe the same when we add a consortia of microorganisms or bacteria in sterile soil. We see that there is an increase compared to the control, but still the microcystins has an impact on the overall of beta glucosidase. The same is observable when we consider the non-sterile soil, and we can see that there is a sharp also difference within the sterile and non-sterile. This is due to some other microorganisms that are, of course, in the cell. And when we add also other microorganisms on the same soil that is not sterile, we see also that there is an increase in activity and also a decrease with microcystins. However, a study by Colbell, when they incubated soil alone without plants, with 72 micrograms of microcystins per kilogram of dry soil, they somehow said or stated that there is an impact of microcystins on soil microbiota, but they didn't find any significant difference of the microcystins on the beta glucosidase. So here, the second enzyme in acetylglucosaminidase, which is an enzyme that catalyzes the degradation or demineralization of both carbon and nitrogen. We see that for this, there is a statistical significance. Either we consider the soil sterile or soil non-sterile with bacteria added, but we can still see that there is a decrease when microcystins are added, but when we add microorganisms, bacteria that can resist or tolerate to microcystins, we see that there is some kind of increase. Again, a study by Colbell, the same study, they did not find the concentration, they did not find a difference when they, it's not the aryl sulfate, it's not the, they did not find the difference, the statistical difference when they consider another enzyme that we are going to get on after. So another enzyme, protease, that is an enzyme that participates in the degradation of proteins in the soil, show that there is also a statistical difference of microcystin impacts on soil. Of course, when we consider the concentration, the highest concentration, 100 micrograms, but when we consider only 50 and 100, there is no statistical difference. And also we can notice, that the addition of microorganisms, the bacteria that resist or tolerate to microcystins, help this protease to increase in soil. Another enzyme, phosphatase, we noticed that also there is a statistical difference of this the microcystins on the overall concentration of this enzyme phosphatase. Also, the study by Kao 200 in 2017 found that when they incubated the soil alone without plants for 30 days, they found that there was a statistical difference when the microcystins is applied to a concentration of 100 micrograms per liter, stating that microcystins has also an impact on this enzyme that is responsible for the release or the mineralization of phosphate in soil. The other enzyme, sulfate aryl sulfatase, also showed a statistical difference in all soils, either with bacteria or not, or either soil sterile or not, showing that these toxins have a great impact on this enzyme. Even if the study by Kobel, when they incubated the soil alone without plants, they did not find any statistical difference for this. This maybe could be due to the short time they incubated, because they did not incubate longer as we have here, like 12 weeks. They only incubated for 56 days. The urease also showed some statistical difference when soil are incubated with or when microcystins are added to soil, also showing that microcystins has an impact on soil. But as we can see here, when we add microbial by bacteria that tolerate microcystins, we see that to certain concentration, like 0 and 50 and 50 and 100, there is no statistical difference, but with a high concentration, the difference is really seen. However, Macau also, the study 2017, when they incubated during 30 days, they did not find a statistical difference. They found differences, but they did not find a statistical significance. So the other enzyme we studied was the dehydrogenase, which is an enzyme that somehow manages the transfer of electron on organic matter, assisting the oxidoreductase reactions, making available the energy necessary to plants or to not to plants, to bacteria, which somehow is known as an index of soil health. So here we notice that there was a statistical difference whether we consider soil that is sterile, or when we add bacteria, or also when we consider alone the soil that is sterile, not sterile, and we add bacteria. We also can see that the increasing, when we add bacteria, we also notice that there is an increase in this enzyme. As we can see also, the study by Kao, they did not notice activity of this enzyme when they have incubated the soil alone for 30 days. So we analyze the total phosphorus in soil to somehow look deeper what happens. And as you can see here, when we have soil that is sterile and without bacteria, we see that the concentration of phosphorus is high, which somehow means that bacteria, because there is no bacteria inside, the phosphorus that is not available to plants still there and not available to plants. Here, I'll remind that this is a total phosphorus, including some phosphorus that is available to plants and other that are not available to plants. And here we can notice that when we add bacteria, when we add the bacteria either to sterile or non-sterile, we notice that the concentration of phosphorus is actually low, which somehow explains the role of phosphorus to the role of bacteria to somehow mineralize the phosphorus, and this phosphorus is taken by the plant to allow or to promote its growth. And we also analyze the amount of carbon that is in soil due to carbon loss due to loss on ignition, and we can see that when we have the sterile treatments, in sterile treatments we have less organic carbon than when we have bacteria, which means that the bacteria can interact with plants and therefore produce some exopolysaccharides or some other organic matter that increases the carbon in soil. The take-home message here in our study is that microcystins are widespread worldwide in different fresh waters, because actually due to global warming there are an increasing of microcystis blooms, which somehow liberates the concentration of microcystins in water, and this water can be used for irrigation, and microbial extracellular enzymes are involved in very important biogeochemical cycling processes that help to assess the health state of a soil. Therefore, the application of microcystins or cyanotoxins on soil reduces the extracellular enzyme activity, which somehow constitutes a problem to plants that are going to be grown on this soil, because the availability of nutrients will somehow be dampened. And as this can be shown by the fact that in sterile soils, or when we have less microorganisms, the activity of the enzymes is lowered. And microcystin intolerant bacteria can somehow increase the activities of these enzymes as long as the other microbiota. So I thank you very much for your attention, and I thank very much the team in Morocco, constituted by different fantastic persons that contributed very, very importantly to this job. And thank you very much.
Thank you, Richard, for this very nice talk on microcystins and their role in the soils. So we'll open up for quick questions for Richard, and then with the remaining time, we can have questions for either of the speakers. So any quick questions for Richard first? Okay, well, we'll open the floor to questions for either Richard or Dr. Tiwari. If anyone has anything that they would like to comment or ask. I have a question for Dr. Tiwari. Do you know if there's any current implementation or use of these diatoms and their products that are being obtained? If there's anyone actively pursuing how their use or exploring more of this?
Thank you for the question. There are a few companies which have commercialized products, which include diatom-based feeds in Europe, and there are also a few companies which are working on or which have commercialized food-based, diatom-based food supplements. So that's there in the market, but it's very scattered, not that common.
Okay.
Thank you.
Thanks. Mina, you have a question?
Yes, thank you both for your great talks. And I have now a question for Archana. And this is still in line, like what Bianca asked, if there is any therapeutic potential application of diatoms, and when we talk about drug delivery and especially targeted drug delivery to a targeted organs or cancer cells. I think I read a lot about it, but if you can tell us more specific about it.
Yeah, thank you. So there are two aspects to it for the drug molecule. One, they can act as precursor for drug molecules because of their metabolites. The different bioactive compound which I had reported in the presentation. Also, they can be excellent drug delivery systems. So we have optimized, I didn't present in this talk, we have optimized with Thiocera, wherein the frustules can act as excellent delivery systems. And their efficiency, non-toxic, the cytotoxicity testing we have done, so that the frustules, because of the pore size, it is very suitable, amicable for absorbing the drug. With cancer-based drugs, the targeted delivery is a challenge. Secondly, the dissolution, solubility of drug is a challenge. So having a delivery system which is eco-friendly and sustainable along with therapeutic values. So delivery system based, diatom-based delivery systems are also one of the much investigated devices, and both ways as source of bioactive therapeutic molecules with drug applications and also as delivery systems. So both ways they can work.
Thank you very much.
Thank you.
Any other questions for Richard or Dr. Tiwari? Richard, what advice would you give to crop growers to prevent the spread of these microcystins through the soil and through the crops?
Thank you very much for the question. Normally, this is an intriguing question because if they use the water, the crude water to irrigate the crops, there is no way to tell that problems are not going to occur. Actually, as a part of my PhD, I am developing an approach that will be available, or let's say not expensive for farmers that use water contaminated by microcystins. And this process, they can use it to eliminate the amount of microcystins to a level that they cannot have problems to plants. Because normally the effects begins to a certain level or a certain concentrations of microcystins. When we're talking about low concentrations, environmental concentrations, they can be easily mitigated by soil microbiota.
Thank you, Richard. So we have a question in the chat for Richard. And based on what you use, the 50 and 100 micrograms per liter of microcystins, and also about the fraction of cyanotoxins you use for your experiment, is it a pure fraction without any source of sugar or anything else that could be used for the bacterial growth?
Thank you for the question. I think this is very interesting because in fact, we take care of that when we were designing the experiments, because we used, of course, a crude extract of microcystins, but before using it, we first used 18C cartridge to somehow purify microcystins as if we are going to quantify the concentrations using HPLC. Yes, here the thing we used, we minimized other components, like to have the minimal as possible, the concentration only of microcystins.
Any other questions for Richard or Dr. Tiwari? Okay, well, if there are no more questions or comments, then thank you both for presenting today, and thank you everyone